So, dear students, good afternoon. I hope my PPT is visible to you and my voice is clearly audible. Yeah. So, in last lecture, we were talking about the biological databases. We talked about the characteristics of the biological databases, features of the biological databases, and in last, we started discussing about the classification of the databases. So the classification of the databases can be classified in many ways. Just like the example I gave you for your class can be classified based on the gender, right? So it may be male and female, based on the religion, based on the type of regularity you are showing in your classroom, right? So likewise, once there is a lot of database in the scientific community, there is a lot of databases are available there. So meanwhile, what we can depends on different criteria. We can classify the databases, right? So databases can be classified based on the data type, which type of data they are, which type of data is being uh, put it in the database, who is maintaining the data, that is based on the maintainer database status, data access, what is the way how the data bases are allows their access, some are making it freely available to all, some are charging some money, means they're proprietary, some are just allowing the access but they do not give the permission to the downloading, right? So there are the different way we discussed. Then the data source, the database design, and the organism is specific database. So these are the classifications that we have already discussed. The main thing was we were started discussing the main classification. That was the primary database and secondary database, and third one was known as composite database. So today we have to finish it up the types of databases, and then we'll start taking the each and every nucleotide databases whichever in your syllabus in detail, one by one. So primary databases, I told you that those databases which are accepting the sequences directly from the researcher. Suppose you are the researcher, so you can submit your sequence to any database. So those databases which allowed the user to directly submit their sequence, just like in the Facebook. Whatever pictures or stories or videos you have, you can directly post on the Facebook, right? So likewise, those databases which allows the user to submit their data in the database, so that if database administrator found it is suitable, then they can make it available for the public use. See, again repeatedly I am giving the example of the Facebook and social media sites. The difference between the Facebook and the biological database, the major difference I let you know, the major difference is that whenever you have any idea or any video or audio or anything, directly you can post on the Facebook, and whatever you are being posting, that will be available to all of your friends within a fraction of minutes, isn't it? So there is no control of Facebook. Facebook talks about the freedom of expression, right to express. So whatever comes in your mind, you can post on the Facebook or in the Twitter or in Instagram, and that will be visible to everybody. But the same thing, once you have any sequence, you have done any kind of the sequencing, and you have done that. Definitely, you also, if you have user ID and password, you can create just like in the Facebook. Until and unless you are not using the user ID and password, you cannot post anything on the Facebook, isn't it? The same way, if you are using any particular database and you create your user ID and password, once you registered yourself, you will get the user ID and password, and with that credential you can log in the database, and you can submit your data to in the prescribed format given by the database, that your information you can post, but that information is not going to be publicly available. That will be withheld with the database administrator. Means if anything comes in my mind, anything rubbish, I can type ATGC something TTC something ATGC I can type, and I annotate it that this is the gene which is responsible for the IQ level in the human being that I claim, and it is going to be publicly available to everybody. Although I falsified the data that don't have any relation at all with the genuinity, right? So that work I have falsified. That work genuinely I have not done it. So do you think if such type of information, if someone posts and it is being made available to the public, does it make any sense? Does bioinformatics or biological databases objective will be fulfilled? No, not at all. Any wrong information or falsified information should not be publicly available. Otherwise, if it is wrong or falsified, there is no meaning of the database. Then now the $1 billion question comes: if database directly not making it publicly available, then how database administrator will verify it whether whatever sequence any user is posting that is right or wrong? Is it possible for database administrator, say for example, let me give you an example like GenBank. So GenBank is maintained by NCBI, National Center for Biotechnology Information, USA, United States of America. Do you think that American scientists will come in your laboratory and they will check manually whether you have done this sequencing or not? Obviously, let me tell you one more fact. What is the speed of increasing of the data in the database? On every 18th month, please make a note of it. On every 18th month, the database size is being doubled. Every 18th month, say for example, today we have 100 TB of the biological data. So after 18th month, it will be around 200 TB of the biological data. So this is the speed of the people are contributing in the biological databases, in the biological data, right? So definitely, if you take the on an average, so if you blink a single time, if you blink in a minute, you know already that we blink between 15 to 20 times. So on every blinking of our eyes means on every minute 15 to 17 people are submitting their sequence to the database. So if in 1 minute 15 sequences are being submitted throughout the globe, then you can calculate how much new data are coming in a year. It's not difficult to calculate, right? Because in 1 minute, suppose 15 sequences are coming. So 15 multiplied by 60. So that will be around 900 sequences per hour, or 900 multiplied by 24, so it will be around 20,000 sequences per day, and all these multiplied by 365 days, so you will come to know that how much now sequencing is going on throughout the world. People are submitting their sequences at very high speed. So it is not possible for any database administrator to visit any scientist laboratory and to verify that whether he have done the sequencing genuinely or he or she have falsified. So obviously then it is the biggest task, the biggest challenge before the researchers, before the database administrator, that whatever sequences are coming, they are genuine or not. So there is a layer of the softwares. They have put the layer of the softwares. Based on that softwares, whatever sequencing you are submitting, that will automatically analyze and they will come up to the conclusion that whatever sequencing you are claiming for, whether that is right or wrong. And they once you submit your sequence, it will take around 1 to 2 months if your sequence is genuine to reflect in the database. Then you may also get the email from the database administrator that this is regarding the query. They want to be very sure that whatever you are claiming for, that is genuine. You may get a phone call even. You may get a phone call, or you may get the mail ID. You have to justify whatever you are claiming for if they ask any kind of the question. If they did not found any kind of the problem, then directly after 1 to 2 months, depends on the load, how much sequence they are getting, they can make it available to the public. Then it will be publicly available. So I think it is quite clear now, right? Now you are getting the idea of the biological database. Once again, I repeat, whatever things comes in your mind that you can post on the Facebook, and that within a fraction of a minute, that will be available to all of your friends in your account. But with the database, whatever sequencing you do, with the help of the user ID and password, whatever you get after registration, that you can post, definitely you will be able to post it, but that is not going to reflect to the researchers until and unless it is verified by the database people. Once it is verified, then after 1 to 2 months, depends how much sequences they are getting, how there is staff and all these things. If they approved, if they think whatever you are claiming for that is correct, then it will be publicly available to you. Alright? So I was talking about the primary database. So primary databases are those databases which directly accept the responses, directly accept their sequences from the researchers. Very simple, and it is archival in nature. Archival in nature means these type of databases, the primary database does not discriminate that if you are working with a virus, you cannot submit your sequence in my database. If you are working with the fungi, you cannot submit. If you are working with the human, you cannot submit. If you are working with the bacteria only, then you can only submit. That is archival in nature. The condition will be very simple. That should be the nucleotide sequence. If you have the nucleotide sequence, irrespective of any organism, you can submit in the primary database. So the main criteria of the primary database, they are archival in nature. I think you people are getting it, right? Then I told you about the secondary databases. Secondary databases are also known as derived databases. Whatever information you are putting forth in the primary databases, if with the help of the software we analyze certain type of information, and we can generate something new, and that whatever findings, new findings you got, that you can make a separate database, so that will be the secondary or derived database. Definitely the secondary and derived databases, not the researcher individually, they are not contributing, but whatever data is available with the primary database, based on that analysis we can make the secondary database. I think it is very clear. So once the, as I already told you that the important feature of the biological database is they are dynamic in nature. So whatever conclusion by the analysis of the primary databases you are making today, maybe after 1 year, a lot of new data will come in the database. So at that time, if you again conduct the experiment, definitely your result may get affected. So secondary databases are also dynamic in nature, and they're fully dependent over the primary databases. If you have any question, you please may ask. I will be very happy to answer you. Then I told you that how to compare between the primary database and secondary database. So that we have already discussed. Now this is the time to discuss the third database, and that is known as composite database. So what composite database is all about? Let me tell you one very simple example. Let me give you an example. I think then it will be very clear about you. Say for example, the Government Science College Wankal, right? Suppose if I want to visit the website of Department of Microbiology, Government Science College Wankal, then do you have any separate website for that? Not at all. Your department don't have must. I think I'm sure about it. Your department don't have separate website, right? You have the website, may have the website as Government Science College Wankal. So in the Government Science College Wankal, there is the main website, and in that main website I may find different departments. Maybe Department of Microbiology, Department of Physics, Department of Chemistry, Department of the Humanity, something, the English and all the subjects, right? So, if I want to visit the Department of Microbiology website, I have to go on the Gujarat and the Government Science College Wankal website. There I may find the list of the departments, right? Likewise, many of the organizations are there. They are maintaining more than one databases, right? More than one databases. So just like I told you, if I want to bitch about the Department of Microbiology, I have to go on the Gujarat, government science college Wankal website. Likewise, there are many organization that make it that maintaining more than one databases. So they are going to be called as a composite database. So in short, what you can say database of databases. Just like let me tell you NCBI. NCBI means National Center for Biotechnology Information. So if you search on the National Center for Biotechnology Information, you will find the hundreds of the databases maintained under the NCBI. So if you open the website as NCBI, you will find the hundreds of the databases. So NCBI is the best example of composite database. Is it alright? Even the GenBank I told you, no, the GenBank database. So GenBank database don't have any different website. If you want to see the GenBank, you can go on the NCBI and GenBank is available under the NCBI. So composite databases are those databases which are the database of databases. Under that databases, you will find the number of databases. So that is simply known as composite database. Alright. So now one more very important concept I would like to discuss here. That if you remember when we started our discussion, even in this particular slide you see the nucleotide, the gen bank, the ENA, the DDBJ. You simply may arise the question. You simply may ask the question to me, sir, why there is three database, the nucleotide, the ENA, and the DDBJ? If all are nucleotide databases, then why we cannot make only one database? Why to maintain three database? So suppose you are the researcher, you are in the search of any particular gene. So you have to search for three databases. You have to go on GenBank, you have to go on the ENA, you have to go on the DDBJ. Meanwhile, if you are the researcher, suppose you have done the sequencing, so you can see researchers are no, they are kind of the selfishness means whatever I have done, I am selfish that I want to make it available to the everybody, right? So for that, so that maximum people can take the benefit of it. So I will upload, I will submit my sequence to the GenBank also, the ENA also, and the DDBJ also. Then unnecessary my time is going to be wasted if I am the researcher, right? And if you are the researcher, you are searching for any particular sequence, you have to search in three databases. So definitely the number of researchers are less, the number of submitters, the people who are contributing in the database are less, and number of researchers are in lakhs. So you are wasting the times of the lakhs of the people. If it is single database, then it might be easy, right? The nucleotide, if I am the researcher, I can submit my sequence to the GenBank. And if you are the researcher who are searching for any particular sequence, you have to search only in one database. There is no need to search in three databases. So that is the problem. That you may arise a question why there is three databases. So let me tell you, the first database was developed by GenBank. The first database developed was GenBank, and that is available with the NCBI. The second nucleotide database was ENA, European Nucleotide Archive, means it is developed by European countries, and the DDBJ was developed by Japan, right? So later, I told you the important feature of the databases are they are autonomous in nature, right? So GenBank is maintained by USA, ENA is maintained by the European country, and DDBJ is maintained by the means Japan, right? So later, whatever concern I raised, then if you are the researcher and you are submitting any kind, if you want to submit any kind of information, then you have to submit your sequence in three databases. And if you are the researcher who are searching for any particular sequence, you also have to search in three sequences, three databases. So a lot of time is going to be wasted. So now the GenBank, ENA, and DDBJ they made one collaboration. Why to maintain three databases? Let's merge all the databases so that the user and the submitter have to see only one database, right? But none one was agree to dissolve their database. USA said, okay, I was the first who made the database, so why I should dissolve my name? You, the ENA people and DDBJ, you dissolve this database with me. Each one have their own perception. So nobody was ready to dissolve the database. So what they thought, let's not dissolve. Let's make it is available in all the databases. Let's make the collaboration. What is the collaboration they can make? And that is known as data sharing collaboration. What data sharing collaboration means, let me tell you, here is the table, the nucleotide sequence, one collaboration was there, International Nucleotide Sequence Database Collaboration. So as per this collaboration, the name of this collaboration is the INSTC and the members are joined by DDBJ and ENA. What they mutually decided, suppose if today I have done any sequencing, right? If today I have done any sequencing and that I submit to the GenBank, for example, then I don't have to, then I don't have to submit the same sequence to the ENA and DDBJ. My job is over. And once the GenBank people will verify that whatever sequence was submitted by the researcher, that is correct, then definitely they are going to make it publicly available, and one copy of that they will submit to the ENA and another copy to the DDBJ. So the same information means the DDBJ, the ENA and GenBank don't have to verify it individually. So if only one verify, like GenBank people verify, that they will send one copy to the ENA and DDBJ, means now your job is reduced. I just submitted, I don't have to submit in 3 databases, right? And as a researcher, if you are searching for any particular sequence, you don't have to search in all 3 databases. Anyone, all the informations are same in any database. You can submit it, you can search in the GenBank, you can search in the ENA, or you can search in the DDBJ, which you are interested in the search. The information available with all the 3 databases are same. So this is the data sharing collaboration. Next is known as protein sequences. There is lot of example I told you for the protein sequences, right? Databases. So there is again they made one collaboration that is known as UniProt consortium. If you submit a protein sequence, if you newly you sequence any particular protein, you submit in one database. After verification, that protein sequence is going to be available in all other databases. Macromolecular structure, if you solve the 3D structure of any particular protein, you can submit in any of the protein structure database and that will be available with other databases also. Likewise, the molecular interaction, the protein identification, the metabolomics data and the genomics and clinical data. So these are the different collaborations are being made so that all the databases can work in the collaboration and there is the less workload and more efficient administration. If you have any question, you are most welcome. You can put your questions in the chat box. So now question arises, how many databases are there in the scientific community? How many databases are available over the internet? So one journal is there, I think you must know about the journal. Whatever new scientific finding you made, that you can publish in the form of the research paper, right? Or in the research paper. So that research paper should be published in the scientific journal. So one journal named as a Nucleic Acid Research. So suppose today I make a database, biological database. So before I make it publicly available, I must write a research paper and I can publish in the Nucleic Acid Research. If that research paper is published in the Nucleic Acid Research of that database will be indexed in the Nucleic Acid Research. So there is the different categories of the database, right? Like nucleotide sequence database, if you are the researcher, if you are working with any particular aspect, you are doing your PhD, after your MSc you will do your PhD, right? And there is the thousands of the databases are available. Then how will you come to know that what particular database I should use for my research? Because thousands of more than 2 to 3,000 databases are available, which is of your use, right? So very simple, you go on the website of the Nucleic Acid Research Very simple. The URL have been given here. Simply put the nucleic acids research in the Google even. It will be navigating to a new page. There you find the database categories list. If you are working with the nucleotide, if you are working with the human genes and disease, so under the category you can find the human genes and disease. It will again show you certain links, and you will find all the databases available and which database suits to you that you may search for. Alright, so this was all about the biological databases. Let me summarize what we have discussed till dated. We talked about some concepts of the bioinformatics. We defined the bioinformatics, the history of the bioinformatics. Then we started our discussion. I told you in bioinformatics there is only major three things that you have to study, that you should understand. The number one was database, number two was algorithm, and number three was software. So we started our discussion on the first part, that is the database. So in database I told you from the nutshell, from the scratch, what database, just a minute. Yep. So from the scratch, I told you what database is all about, what are the building blocks of the database, what is the DBMS, what is the benefit of the database, what are the limitations of the database that we already talked about. Then we started discussing about the biological databases. Why do we need the biological databases? What are the characteristic features of the biological databases? And then then finally started a discussion over the classification of the database. There is a lot of way we can classify the database. Then in that the measure was the primary database and the secondary database and the composite database. I also talked about some examples of the primary, secondary and composite database. Then I talked about the data sharing collaborations, means why there is more than one database for a single purpose. If it is nucleotide database like GenBank, then why other ENA and DDBJ are existing? So for that one, I told you why we need the collaborations, and collaborations is always good. So certain collaborations names I also told you. So now, as I can see in your syllabus, you have to go for detailed study of two databases. The number one database you have to study about the EMBL, and number two is DDBJ. So now I am going to teach you the EMBL and the DDBJ at the end of the lecture today. Now you have enough background about the biological databases. How many biological databases are there, right? What are the different classifications of that? So at the end of the lecture, I am going to share some of the videos. That videos you have to watch by your own. That is actually the recorded video of for the practical session. So someone was asking yesterday that how to use the biological databases. So if you if you watch those videos carefully, you will get the answer that how to use the biological databases for your research and how many ways you can access it. Okay. So at the end of the lecture, I will share in the group some of the tutorials how to use the biological databases, right? So that practical portions you see later on, but before that, let me teach you the details about the ENA and the DDBJ. If time permit, I also will teach the GenBank. although that is not in your syllabus. Let it open. So the primary database, as I already told you, there is three primary databases mainly for the nucleotide: the DDBJ, the GenBank. It is written here as NCBI, but since uh, for the for the INSTC, no, we consider it as the GenBank and ENA. And these are three databases are collaborated with one uh, these all three databases are connected with one collaboration, and that is known as INSTC, International Nucleotide Sequence Database Collaborations. The benefit of the INSTC: suppose you are the researcher, you have done any kind of a sequencing, you submit to the DDBJ. Once DDBJ people will verify it, they will send one copy to the GenBank and another to the ENA. So you don't have to waste your time to submit to other two databases. And researchers can save their time. So the same information will not be verified by other two databases. So that there's a lot of time and effort can be saved by the scientific community. Okay, so primary database like nucleotide, I talked about three examples: the ENA, the GenBank, the DDBJ. So ENA, the full form of ENA is European Nucleotide Archive, and the older name was EMBL, European Molecular Biology Laboratory. The GenBank and DDBJ, DNA Data Bank of Japan. For the protein, you can say the PIR, the MIS, the SWISPROT, the Trimble, the NRL3D. Obviously, we are not going to read all these databases. We are going to discuss only those databases which are in your syllabus. All right. So, if you want to see, if you want to visit all these three databases, the link have been given in PPT, the EMBL for the GenBank, for the DDBJ, and they together constitute the INSTC, International Nucleotide Sequence Database Collaborations. So, if we talk about the GenBank, let's talk about one by one. The GenBank. What is GenBank actually? It is an annotated collection of all publicly available nucleotide and proteins. See here the contradiction. Till dated, I talked about that GenBank is nucleotide database. Then you see one contradiction. Here it is written publicly available nucleotide and proteins. Okay. Although in the GenBank, major form, major sequences nucleotide database, nucleotide nucleotide sequences, but protein sequences are also there. But since the database covers majorly is specialized in the nucleotide database, so roughly we can say it is nucleotide database, but it doesn't mean that it don't have the protein sequence. Protein sequences also can be found in the GenBank. The same concept will be applicable over the DDBJ and the ENA. So protein related information we may also find in these databases, but since they are specialized for the nucleotide, that's why they are being called as a nucleotide databases. Alright, so you should not have any confusion. So for example, in your examination, if it is asked that which type of information is being hosted, being available in the GenBank, EMBL and DDBJ, so if option comes only nucleotide, only protein, your answer will be only nucleotide. And if options are like both the nucleotide and protein, then you have to go with both nucleotide and protein. So roughly, we can say GenBank is the nucleotide database, and it was set up in 1979 at the LNL Las Almas and maintained since 1992. NCBI, I told you that now GenBank is maintained by NCBI, National Center for Biotechnology Information, and that is the example of one composite database. And here you can say the URL, the internet address you may find. If you talk about the EMBL, so it is again an annotated collection of all publicly available nucleotide and protein sequence. You may ask the question, what does annotation means? Annotation means very simple. If you do any kind of the sequencing today, so whatever ATGC you get back with the help of the sequencer, that ATGC only you are not going to submit with the database, right? Some value addition you will also put in that. You have to annotate. In annotation, what you have to put, like whatever sequence is there, whether that is coding sequence or non-coding, it is intron or no, intron or exon, where is the promoter, where is the operator, where is the enhancer, where is the silencer. So all the values you can add in that. You can tell that starting from this basis to this base it is promoter, starting from this to this it is non-coding, starting from this to this it is coding. So whatever extra things you can put after analysis, that is known as annotation. So what actually you can see in the EMBL, it is an annotated collection of all publicly, whatever all publicly available nucleotide databases are there, they are available along with their annotation. So it was started 1 year later after this journal, created in 1980 at the European Molecular Biology Laboratory in Heidelberg, Germany, and since 1994, now it is maintained by EBI. EBI stands for, somewhere you can write down the notes also, because it is not possible that everything I can put on the PPT, right? So EBI stands for European Bioinformatics Institute. European Bioinformatics Institute. So now one more thing, that a composite database example is EBI. So under the EBI, if you open the EBI, European Bioinformatics Institute website, you may find the number of databases. So in examination, you also may write the example of the composite database are two, the SCBI and the EBI, although all other databases examples are there. And you can find the URL of to assess the EMBL. The third is DNA, DDBJ, DNA Data Bank of Japan. The definition is same for all. The annotated collection of all publicly available nucleotide and protein sequences. It was started in 1984 at the National Institute of Genetics in Mishima, Japan, and is still maintained in this institute, a team led by Takashi Gojabori, and this is the URL to access the DNA Data Bank of Japan. Fortunately, if you want to access the DDBJ, you don't have to go for any composite database. The direct URL you may find the ddbj.nij.ac.jp. Directly, if you click on that, the direct database you will find, right? So other databases are there. We are not going to discuss all the databases because I told you that name of the databases are in the thousands. So what I do now, before I go much detail about the ENA and DDBJ, let me give you some practical hands to you so that you get the idea. Okay. So as of now, I am stopping my session and I am going to share the video link, the practical link. So before tomorrow, you just please have a look on it, do the practical as instructed in the video, then the concept of the biological databases will be very clear in your mind. And once after that, if I explain the theory, the things will be very easy to you, right? So please make sure that before that, watched all those videos which I am going to share in your group. Then tomorrow, to understand the concepts will be very easy. So see you tomorrow at same time. Till then, stay safe, take care.